Hello, my name is Angelica Sanchez and I will present the work Genome Sequence of Staphylococcus epidermidis strain 252 from the rhizosphere of an endemic plant of the Iron Quadrangle. Mimosa calodendron is a species of leguminous plant endemic to the rupestриan fields of the Brazilian Iron Quadrangle region. However, only recently the cultivable microbiota associated with this plant has been studied. Also, the Iron Quadrangle displays extreme environmental conditions for microbial communities, including a soil rich in metals, poor in nitrogen and phosphate, high UV incidence, and temperature ranges. Despite these adverse conditions, the surviving plants are highly adapted, and one of the reasons of success of plant adaptation is due to the plant-microbe interactions that through complex networks of interactions can promote plant growth and suppress plant diseases. To study these mechanisms provides insights on the ecology of plant-microbe interactions and the biotechnological potential of the cultivable microbiota associated to these plants for the agriculture and environment. The objective of this work was to sequence the complete genome of Staphylococcus epidermidis strain 252, isolated from the roots of Mimosa calodendron, to correlate the gene repertoire with its adaptation to the soil of the Iron Quadrangle, and to compare this genome with other 4 species of S. epidermidis isolated from rice plant, looking for specificities that allow the isolate to adapt and become endophytic to Mimosa calodendron. For that, we performed the DNA sequencing with the Illumina MySeq. All comparative analysis involving metabolic pathways and cellular processes were done within the RAS and BRIG platform. This strain 252 chromosome has over 2.5 billion base pairs. Its annotation resulted in 2,422 protein coding genes. The strain 252 genome has 2 phage insertions and 8 genomic islands. Comparative analysis with other 4 complete genomes of S. epidermidis associated to plants revealed a core genome of 3,668 genes and 140 unique genes. Unique genes present in one of the strain 252 genomic island encodes a complete staphyloxanthine biosynthesis pathway. Staphyloxanthine is an orange-red carotenoid, and it's found in Staphylococcus aureus, where it acts against oxidative stress, being one of its virulence factors. And among the strain 252 genes, we highlight that the isolate has the complete cluster of genes associated with the synthesis of type 7 secretion systems, which is present in gram-positive bacteria, including important pathogens such as Bacillus anthracis, Mycobacterium tuberculosis, and Staphylococcus aureus. The comparative analysis of the subsystems classification of RASs for the plant-related S. epidermidis genomes shows a repertoire of abiotic and biotic stress resistance and genes related to plant growth promotion. The 252 strand presented 3 genes related to the degradation of aromatic compounds. It was found that the strain has a repertoire of genes associated with metal internalization and osmotic and oxidative stress, and also DNA repair. We also found genes that are associated with phosphorometabolisms and transport, a complete cluster of genes for the denitrification pathways, genes associated with the conversion of antranilate to tryptophan, and genes associated with the transport of the siderophore anthracylene. Classically, Staphylococcus epidermidis is associated with animal epithelial tissue, only recently described as a plant-associated. In this work, for the first time, an S. epidermidis strain 252 was identified in an endemic plant of the Iron Quadrangle. This finding shows that this isolate is adapted to different biotic and abiotic stress situations imposed by extreme conditions associated with ferruginous fields into the roots of Mimosa calodendron. We would like to thank the funding agencies, the Federal University of Ouro Preto, the Biological Society's Research Nucleus, and the Biotechnology Graduation Program, the co-authors of these works and members of the DNA and the Genome and Bacteria Environment Interaction Labs, and the organizing committee of the Brazilian Society of Genetic Congress. Thank you all for your attention.